Hello friends, welcome. So in today's session, we will talk about nerve impulse generation and conduction. So let's first talk about the nerve impulse. What is nerve impulse? Before that, we knew that neurons or nerve cells are the basic unit of nervous system. What is nervous system? If we talk about the nervous system, so nervous systems is considered as the command center of our body. It is the control center of our body. So by the way, the body is controlled by the nervous system and also by endocrine system. So in nervous system, neurons are the key player which are essential for the communication and the control. So if further, we also knew that the neurons has two part mainly. If we talk about nerve cells or neurons, so nerve cells has two main part. As we knew, it has cell body and cell process. So also further we knew that the cell process has two zone, that is the dendron, and also sometimes the branches are there which are called as the dendrites and axon. We knew that the dendrons are those processes which receive nerve impulse and transmit it towards the cell body, and axons are the process which receives this nerve impulse and transmits the nerve impulse away from the cell body, or it can directly, or it is involved in the transmission of nerve impulse from cell body to axon, and it can also used for communication between the neurons. So these are the basic detail as we know so far. So the idea is that how the neurons generate nerve impulse, and how the conduction of these nerve impulse are taking place. So, so let's start with the generation process. Like, let's take a situation when there is no nerve impulse transmission is seen from a neuron, right? So those state, the state when there is no nerve impulse transmission is seen, the state is known as the resting state. That is, there is no work at all is happening from the neuron. Basically we knew that the neurons or nerve cells are basically cells, so a lot of metabolic activities are going there every time, every single time. But the main work of the neuron is, as we know, that since it is part of nervous system, the role, major role is to transmit nerve impulse, to transmit the electrical signal, and playing a role in controlling the body. So what is happening? So let's start with where there is no nerve impulse transmission is there, and the state is known as the resting stage, right? Resting stage when no nerve impulse transmission is there, right? No nerve impulse transmission is there. So at this state, since there is no nerve impulse transmission is there, this stage is known as resting state. So what is happening there? As resting state, what is happening there? So consider the situation when the neurons are in resting stage. Membranes, by analyzing the membranes, the physiology of the membranes, when we look deeper inside the neuronal membrane, what we look that there are differential distribution of some ions, right? So when we closely monitor the membrane of the neuron during resting stage, we see that the charges or some ions are differentially distributed. So why I'm saying this, that when we look, we will see that outside of the membrane, outside of this membrane, what we see that the sodium charge is high, sodium ion is high. Along with, we will also find that chloride ion is also there. So this is the actually this is the differential distribution where we are considering it as the overall concentration is high outside of the neuron, and it is sodium ion which is high outside, and inside of the neuron the potassium ion is relatively high. And also we will find that some of the negative charge protein is high inside. Overall distribution of this sodium and potassium ion along with chloride and negatively charged proteins, the charge, overall the charge appears outside is high. We consider it as the positive charge outside because of the differential distribution of dominance of sodium ion outside, and inside it is high, potassium ion is high, but relatively the concentration is not up to the mark. That's why it is considered as the low with respect to the outside. So this distribution of the charge creates a partial gradient of charge as compared to inside the outside charge is high. Now the differential distribution of this charge makes it a polarized state. The membrane is now called as the polarized. The term pole, we know that in our earlier classes, or when we move to next class in physics, we will see that if there are two different charges, positive charges and negative charges separated by certain distance, these charges are called as dipole. Right, since the two type of charge, dipole, pole, that is these two charges are forming pole. Also in magnet we have seen, studied that magnet has two different poles, north pole and south pole. So with the analogous similarity from this pole, the membrane, since most of the time membrane is showing that nature, differential nature of membrane, showing outside positive inside negative due to the relative distribution of charge, this state is called as the polarized state. That is, membrane is in polarized state. At this stage, the this differential distribution of charge creates a gradient which is approximately minus 70 millivolt. So at polarized state, that is during resting stage, when there is no nerve impulse transmission is happening, membrane is polarized and the voltage across membrane is minus 70 millivolt. So this is the overall physiology during this resting stage. But the question is that how this polarization of membrane is maintained, how this polarization of membrane is maintained, because we know that cells are metabolically active and it requires different metabolic inputs, and also it releases some metabolic waste and something like that. So we know that, so the maintenance of this resting state or the polarized state is done by with the help of some of the channels present in the membrane and some of the pumps which are also present in the membrane. So before moving to next, we will talk uh, more details about the channels and the pumps. So like if we have the neuronal membrane, we have some pumps and some channels. So what is the role of pumps and what is the role of these channels? So pumps are basically sodium ion, potassium ion pumps. For the activity of pumps, these sodium and potassium ion pumps, for its activity, it requires input of energy, which is in form of ATP and energy input. Right? So when pumps work, sodium potassium pumps work, it requires energy input in forms of ATP, and that input of energy drives the pump for its action. And also, what is okay, so now what is channel? So channels are basically also membrane, part of membrane, which are found in membrane basically, and it acts as the charge carrier, or not, I should not call it charge carrier. Basically channels are the media through which charge or ions are moving inside or outside basically. So the channels provide the passage for the migration or diffusion of the charge. Channels doesn't require ATP or energy. Basically, when we talk about the channels, does it require energy or ATP input? No. Channels are basically, when we talk in context of NEET level up to our level, we say there are two types of channel: leaky channels and voltage gated channels. Voltage gated channels. Remember, the channels doesn't require energy input. So some of these channels, which are leaky channels, continuously leaks ions or charge where there is gradient. Like along the gradient, concentration, electrochemical gradient, charges or ions are leaking. That is, it may leak outside and it may leak from outside to inside where there is more electrochemical gradient seen. Also, some of the special channels which are not always open, but when approximate voltage or membrane potential is changed, at that time some of the channels open. Since it is opening in influence of membrane potential change, that's why it is known as voltage gated channel. So leaky channels always open and leaks ions either outside or inside. But the voltage gated channels only open under influence of change in membrane potential. Change in membrane potential. So these are the basics of pump and channel which are found in the neuronal membrane. Now, these have a very essential role in maintaining the resting stage of the neuron membrane. So at resting stage, since it is minus 70 millivolt across the neuronal membrane, and why it is minus or negative 70 millivolt? Because the outside is positive and inside is negative. That's why it is considered as minus 70 millivolt. Pumps are those of two types, that is basically sodium and potassium pumps, which which essentially carrying out 3 sodium outside of the neuron membrane. It is carrying 3 sodium outside of the neuron membrane at the cost of 2 potassium inside of the neuron, and that's why it is called as sodium potassium pump, because it is carrying out 3 sodium outside and 2 potassium inside. It requires energy input, and overall its activity is essential for maintaining the polarized state of the membrane at resting stage. And also this stage is maintained by the leaky channels. So this stage is maintained by the leaky channels. Leaky channels also leak where there is excess charge. Where there is excess charge, it leaks in opposite side to maintain minus 70 millivolt. And most frequently it is seen that the leaky channels during resting stage leaks potassium ion outside of the axonal membrane. So this is overall state of the resting stage when neurons are not carrying any nerve impulse or electric impulse. So how generation of the electric impulse is happening? Let's take another situation when there is stimulus. When there is a stimulus, or when uh, some previous neuron is carrying out the nerve impulse and it is approaching to the next neuron. So in that case, how the generation of nerve impulse is happening? So if there is a stimulus, how it is driving the generation of nerve impulse? So during a resting stage, we knew that outside is positive and inside is negative. Outside is positive and inside is negative. This is the situation when membrane is not carrying any nerve impulse, that is resting membrane potential. So if there is a stimulus approaching to the dendron or dendrites, if there is a stimulus, if there is a stimulus, the presence of a stimulus leads some local channels to open, or if there is nerve impulse, it opens some channels due to change in membrane potential, or simply it can say that the stimulus or the nerve impulse causes the local channels to open, which leads sodium ion influx. So in that case, where there is stimulus, what is happening, that the since outside is positive and sodium is moving inside, sodium ion is moving inside, the membrane potential from minus 70 millivolt, due to minus 70 millivolt, because of that inside is more negative, right? So since from outside sodium ion is moving inside, it is approaching to positive side, right? It is approaching from minus 70 millivolt to positive side, that is minus 70, minus 65, minus 60. Why? Again, because of dominance of positive charge inside. At this local site, what is happening? Sodium ion is moving inside, and due to this, the inside is going to be positive, and it is now increasing this membrane potential from minus 70 millivolt to minus 60, minus 50, minus 40, that is so on. But there is a glitch. What is the glitch? Actually, the change in membrane potential due to stimulus, the change in membrane potential due to a stimulus is initiating the sodium ion influx. But if the stimulus is strong enough that the continuously sodium ion is moving inside and leading to change in membrane potential from minus 70 millivolt to minus 55 millivolt approx. right? This is the scene when most of, due to change in membrane potential from minus 70 millivolt to minus 55 millivolt, at this point, this is the stage when most of the sodium ion channel voltage gated sodium ion channel opens up, and also due to at this potential, at this membrane potential minus 55 millivolt, potassium channel is also opening, starts opening. So the point is that a stimulus is, if a strong enough to take over this minus 70 millivolt to minus 55 millivolt, then this will lead to opening of the voltage gated sodium or potassium ion channel, right? At this stage, sodium ion voltage gated channel and potassium ion voltage gated channel will open up, not open up, begins opening, right? Begins its opening. The point is that, but if the stimulus is not strong enough, if the stimulus is not strong enough, it doesn't lead the membrane potential from minus 70 to reach minus 55 millivolt. That is, if the membrane potential is not reaching to minus 55 millivolt, that is, if it is minus 65 millivolt, then this will not lead the opening of the sodium or potassium voltage gated channel. And since the voltage gated channel is not enough, that is, it will not leads further entry of the sodium ion inside. So this is the threshold, where it leads the sodium and potassium voltage gated channel opening. That's why all stimulus are not, only those stimulus are effective which are which is initiating this process of opening of sodium and potassium voltage gated channel, and that's why it is called as all and none. So now let's take the situation when if the membrane potential is reaching the threshold, that is minus 55 millivolts. So what is happening there? It is leading the sodium and potassium voltage gated channel opening. So once again, if we consider it as the sodium ion voltage gated channel is opening, this is the fast process. That is, the opening of sodium ion voltage gated channel is fast process, and it is slow process. It is slow process. Now the situation is that at its minus 55 millivolt, the voltage gated sodium ion channel is opening. Voltage gated sodium ion channel is opening and leading sodium ion inside. Further, it is leading more and more sodium ion inside, and that's why the membrane potential at that particular state is changing, become inside become more positive and outside now become negative. But the stage is ahead of this, outside is still positive because of the localized voltage gated sodium or potassium ion channel opens. Since it is due to fast opening of sodium ion voltage gated channel, the sodium ion is going inside. Sodium ion is going inside. It is not due to the potassium ion channel. And at this stage only, the membrane potential is changed from minus 70 to minus 55, and now inside become positive, and at this stage the membrane potential is plus 30 millivolt. This is the stage membrane become depolarized, right? Why depolarized? Because already we have given the term polarized membrane when outside is positive and inside is negative, but since it is reversing the situation, outside is negative now and inside is positive now, that's why the situation is that then the membrane is now depolarized. It becomes plus 30 millivolt. What is happening now, that at this stage when outside is negative, so the charge is jumped from this positive to negative, and so on, A to B, B to C, C to D, B new, so far that from this charge jumping, it become negative and positive, it is positive and negative. So further what will happen, that this negative will again become positive, and this positive will now become negative, this positive will now become negative, right? Like this, because of jumping, this become negative to positive and positive to negative. So the membrane potential is changed at this site in later stage, and this side will be depolarized then in next step. So this is the case of depolarization. So this is the case of depolarization. This is the case of depolarization, right? So we have gone so far from polarization to depolarization. Now the case is that after conduction of this nerve impulse from site A to B, B to C, C to D, now the stage is that membrane cannot be always in depolarized state, because since once the neuron is depolarized state, it cannot generate another nerve impulse. So to generate another nerve impulse, the neuron must have to reach at the repolarization, that is, it has to attain minus 70 millivolt, that is RMP, so that it can now lead again generation of another nerve impulse. So how it is happening? So we have discussed here that the opening of this sodium ion voltage gated channel is fast process. So as soon as this voltage gated ion channel due to voltage gated ion channel, sodium ion voltage gated ion channel, the membrane potential become plus 30 millivolt. This sodium ion voltage gated channel will close now. That is that is that is the case that this sodium ion VGC is closed now. Since potassium ion VGC is slow opening, showing slow opening, so at this stage now the time when potassium ion voltage gated channel is open. So at this stage the K plus VGC opened. So remember, it is very unique biology that it start at simultaneously with sodium ion VGC, but it opens fully when it is plus 30 millivolt, that is delayed opening. But the sodium ion BGC, so fast opening and fast closing. So sodium ion BGC is closed now, and potassium ion BGC is opened now. So now since potassium ion BGC is opened, so at this stage what is happening, that we have seen that in this stage, since it is outside is negative, inside is positive, and it is leading the potassium ion VGC open, that is K plus VGC is now open. So now charge has to move, that is the potassium has to move now, not sodium, because sodium ion VGC is closed. So it must be potassium ion which has to move, because of this K plus VGC is opened. Since K plus VGC is opened, so only K plus will allowed to move. So K plus will move in which direction? That determines the electrochemical gradient of that K plus. So inside is positive and outside is negative, but membrane has to maintain outside positive and inside negative. So how it is possible? So it is possible only that this membrane, along this membrane only K plus is that is positive charge, since positive charge is allowed to move, and it is only potassium. So if potassium is leaving from inside to outside, then positive charge will move from inside to outside, and inside become negative. The same case is happening here. Potassium ion, due to opening of this potassium ion BGC, will leave this membrane from inside to outside. That is, from this it is leaving outside, from membrane it is leaving outside. So this is the case when outside is now going to be positive, outside is now going to be positive. And why? Because of the movement of this potassium ion from inside to outside, from inside to outside. So outside is going to be positive, outside is going to be positive. The case, this is the case when membrane is going to repolarized. Membrane is going to repolarized, as we know that the polarization is the case when outside is positive and inside is negative. So this is the case when membrane is going to be repolarized. But the catch is that as soon as from minus 30 millivolt it is reaching to plus 30 millivolt, it is reaching to minus 70 millivolt, the signal is sent, and neurons try to stop this K plus BGC. K plus BGC, neurons try to stop this K plus BGC, that is a stop signal for K plus BGC. But we know, what we know, that this K plus BGC shows slow opening and slow closing. This is the case. So at this stage when minus 70 millivolt, the K plus BGC shall remain closed. But what is happening? So it is happening that due to the slow closing, delayed closing, the potassium is again leaking outside. That is the outside going to be more and more positive, more and more positive, more and more positive than inside. And this is the case when it's since it should remain at minus 70 millivolt, but it is approaching minus 90 millivolt, minus 100, minus 110, sometimes it reaches minus 110. So membrane potential changes to minus 110. So the case is that the membrane should be repolarized. The case is the membrane should repolarize to minus 70 millivolt, but it is repolarizing to minus 90 and minus 100 millivolt. And at this scenario, the membrane is called as hyperpolarized. What? The case is hyperpolarized membrane. Hyperpolarized state when the membrane potential is becoming more and more positive outside and less negative inside. The case is that relatively called hyperpolarized state of the membrane. But again there is catch. Membrane should not be at minus 90 millivolt. Membrane should not be at minus 110 millivolt. So it must to reach that, it must be there at minus 70 millivolt. So now who is playing the role? So since outside is positive, it try to maintain at minus 70 millivolts, and which is going to play a role? The leaky channel and sodium potassium pump. These two combinedly affect this state and try to maintain this membrane potential from minus 70 to minus 90 millivolt to minus 70 millivolt. And the case that leaky channel is not now flowing through outside. Actually it is flowing from outside to inside. From outside to inside, it is carrying the potassium ion from outside to inside. So the flow is now from outside to inside when it has to reach from minus 90 millivolt to minus 70 millivolt, or from hyperpolarized state to resting membrane potential state. So this is the overall stage, the generation and conduction of nerve impulse, and how the membrane potential is maintained from the resting stage to depolarized state, and then again hyperpolarized state to the polarized state, that at the resting membrane potential state. So this is the overall case and overall the mechanism of the generation and conduction of the membrane potential. Now next point is that as soon as the membrane potential from dendrites the membrane potential or the nerve impulse is moving towards the cell body, and from cell body it is taking away by axon to the axon terminal or axon end. So how the axon end is now used or transmit this nerve impulse from pre-neuron to post-neuron, right? That is from first neuron to second neuron, that is the from axon end to dendrites of the next neuron. The case is that we'll discuss here. So the case is that the membrane potential is now changed, that is nerve impulse is now reaching from the dendrites to cell body and to axon end. This is the axon end, axon knob of pre synaptic. Why it is synaptic? Let me be clear with this. So this is the state when the axon end of first neuron is here, and the stage when the dendrites of the second neuron is here connected. Dendrites of the second neuron is here connected. So axon of first neuron and dendrites of second neuron, this is the dendrite receptor of the dendron. Since it is connected here, there is a space. This is the junction. Overall, this is the junction. Overall, this is the junction. Neuronal junction. It is called as neuronal junction. Why neuronal junction? Uh, because of the axon of first and the dendron of the second. Since both of these axon and dendrons are meeting, this is the neuronal junction, and it is called as synapse. It is called as synapse, and the space is called as synaptic cleft. The space, the empty space between this is called as synaptic cleft. So this is the overall the anatomy or at the junction of the neuron when two neuron meets. So now the case is that if the neuron or nerve impulse, if the nerve impulse is reaching from at this at this axon end. So what is happening there? So since there should be outside positive, inside negative at this resting state, but the nerve impulse is reaching here, causing the inside negative and outside negative and inside positive. The change in membrane potential causes opening of voltage gated calcium ion channel. Voltage gated calcium ion channel. The voltage gated calcium ion channel since opens at this stage, it is leading the calcium ion influx. Calcium ion influx from outside, the calcium is reaching inside. From outside, the calcium is reaching inside. The deposition or increasing concentration of calcium ion inside, now it fuses with the vesicles. What are these vesicles? These vesicles are the structure which has the neurotransmitter, like we know that acetylcholine, GABA, etc., etc. Glutamine sometimes also plays at the neurotransmitter. So this is the vesicles which is having this neurotransmitter. So when the concentration of calcium ion increased inside, it fuses with the vesicles, delivers actually this fusion of the vesicles with the axon terminal is seen. The vesicles is going to fuse now with the axon terminal, leading to release of the neurotransmitters in the synaptic cleft. In this synaptic cleft, that is the neurotransmitter concentration is going to increase here. Neurotransmitter concentration is going to increase here. Now this neurotransmitters from this space is reaching towards the receptor of the dendron. So at the particular receptor, it tries to fuse here. It leads to generation of the localized, actually it leads to generation of the nerve impulse by localized opening of sodium ion voltage gated channel, as we have discussed in the initial stage when the membrane is reaching from polarized state to depolarized state. The initial change in membrane potential is due to this sodium ion influx. So when this neurotransmitter is approaching to postsynaptic membrane, the dendron receptors, and it is leading the sodium ion influx, it is changing the membrane potential at this restricted space, and further, since once it is reaching to threshold minus 55 millivolt, further it will lead to opening of more and more number of sodium ion voltage gated channel and will lead to more influx of sodium ion, and from this state it will lead to the conduction of the nerve impulse in next neuron now. So this is the overall anatomy and physiology uh, of the synapse and the nerve impulse transmission across neurons. Next point is that so what kind of synapse are there? So we have to read two types of synapse, that is there are two types of synapse. So there are two types of synapse: electrical synapse and chemical synapse. There are two types of synapse: electrical synapse and chemical synapse. In case of electrical synapse, the axon of the presynaptic neuron is in very close proximity to the dendron of postsynaptic neuron. It is forming a channel with the help of some of the protein known as connexon. So you don't have to really bother about. So you just know that the in electrical synapse, these neurons, these axon and dendron are in close proximity. That is, they are located very closely, and they form a channel, and through this channel the direct input of the ions, the direct flow of ions from this channel, sodium ion is flowing from pre to post channels, post neurons actually, through this channel. So the ion flow is taking place directly from presynaptic neuron to postsynaptic neuron. So in this case, if the impulse is generated, it will lead to flow from one neuron to another neuron. There it is directly, it is happening there, that is, it will happen continuously. There is no modulation, there is no restriction. If once the nerve impulse is flowing from one neuron, it will definitely leads to flow inside next neuron. So there is no modulation here actually. But in chemical synapse, so what is happening here, the neurons, pre and post synaptic neurons, are little bit on having some gaps. The chemicals are secreted from axon to synaptic cleft, and it is reaching to the dendron of the next neuron, and based on the variation of the chemicals, that is, which is secreting as neurotransmitter. So listen, this is the very important point, that in electrical synapse there is only flow of sodium ion, because of to change the membrane potential, sodium ion is flowing only. But here the storage of the chemicals in axon may vary, that it may be sometime different, it may sometime A, sometime B, sometimes C. So there are variety, there are the possibility that the vesicle contains numerous type of neurotransmitter, and also the chance is that the receptor is also different here. So only allow those a kind of chemicals which are essential to bind, and not every kind of chemicals to bind it. So there is variety in the vesicle and there is variety in the receptor here. So this is the possibility. So based on that, it can modulate. That is, if sometime the neurotransmitter which are excitatory in nature, so if neurotransmitter which are excitatory in nature are secreted here, so it can excite next neuron to conduct the nerve impulse now from this neuron to next, from next to next, from next to next. But sometimes it may also happen that the nerve impulse is not necessary, the signal is not necessary to carry out. So that's why there is block, there should be block, and the nature of neurotransmitter will decide the blocking. So if there is blocking signal, the neurotransmitters are secreted, and these are the inhibitory type of neurotransmitter. So the possibility is that the vesicle contains sometime excitatory type of neurotransmitter and inhibitory type of neurotransmitter, so that modulation can happen at that synapse site. Our human system is more advanced, not more advanced, most advanced system in all the animals and all the living organisms. So the modulation is also necessary because of the diversity in the axon, the diversity in the response, in diversity in behavior. So that sometimes rage, sometimes happiness, sometimes something, so sometimes different mood. So there is the diversity in so diverse possibilities there. That's why our human system is not carrying the electrical synapse. Most of our human system is carrying and having this chemical synapse to modulate the nerve impulse, to modulate the nerve signals. So remember, our human system is mostly having this chemical synapse, not electrical synapse. So this is all about the generation and conduction of the nerve impulse. Thank you. We'll discuss later more.